Yeah.
Okay. All right, let's start. Hi, everyone. Thank you so much for joining this live talk with Melanie Schürz. Melanie is a part of the leading Voltzmann Institute of Nanovesicular Precision Medicine at Paris London University of Salzburg. After the PhD focus on oxidative stress in the liver, Melanie moved as a postdoctoral research to the lab of Nicole Meiser-Kober, where she worked within the EVTT Center, a translational research center for extracellular vesicles, with particular interest in the development of EV-based therapeutics and drug delivery applications. In this context, the lab also developed advanced imaging-based technologies for single vesicle characterization and quantification in different in vitro and ex vivo models. 1 year ago, Melanie joined the newly founded Ludwig Boltzmann Institute for Nanovesicular Precision and Medicine, where she currently leads the research program line Nanovesicular Technologies. Melanie is going to present us with a talk about EVAnalyzer, from single cell physical characterization to EV quantification in diverse in vitro and ex vivo applications. I'm joining and moderating with Heather. She's going to be the co-host. And if you guys have any questions, you can put it in the chat. And in the end, we will read the questions in the chat, or you can raise your hand if you want to ask Melanie a question. So Melanie, go ahead.
Thanks a lot for the introduction, and also thanks a lot for the invitation to give a talk here. I want today tell you a little bit about, first of all, the Institute, the Ludwig Boltzmann Institute, where I'm working at, what we are doing. And then I want to focus on an application that we have developed, which we called EVAnalyzer, and how we and also you can use it to do single vesicle imaging, single vesicle characterization, and also how to use it to track EVs in different cellular or in vivo settings. So as was already said, I am part of the Ludwig Boltzmann Institute for Nanovesicular Precision Medicine, which is placed at Salzburg in Austria. And so the team is really focusing on these round vesicles. So our mission is to translate this small new world of nanovesicles into precision medicine. So the goal is really to bring extracellular vesicles into the clinics, and there are approximately 40 scientists from 15 different nations working on this goal. So we have a very broad range of expertises here. We ranging from organic chemistry to molecular biology, cell biology. We also have some people who do programming here, and also people that are involved and focus on medical affairs, regulatory affairs. So it's a very broad and nice team. The institute is divided into 4 different research lines. The first research line is led by Nicole Meissner-Kober, who is also the director of the institute. And within her line, we are focusing on nanovesicular-based drug delivery. So how can we use these vesicles to load it with drugs and then go for targeted delivery of active pharmaceutical ingredients. And here we mainly work with EVs from edible sources, like, for example, milk or lemon, and sources where we can also go into scale manufacturing. The second line, which is led by Mario Gimona, is focused on nanovesicular therapeutics. So it's really about the inert and the properties of the vesicles themselves. He works with stem cell EVs for applications in, for example, tissue regeneration. The third line, as you have already heard, is focusing on technologies. So here we have already built quite a fundament with the previous projects like the EVTT, where we have set up different technologies, how to isolate these vesicles, how to characterize them, and how to track them in different applications. And the fourth line, which is led by Eva Rode, is focusing on science, medical, public liaison, and open innovation in science. So how can we pave the way for vesicles into the clinics by interacting with patients, with caretakers, and also with regulatory affairs and with regulations. Today in my talk, I will focus a lot on the nanovesicular technologies, of course, because it's my expertise, and also it's where we have developed EVAnalyzer. So nanovesicular technologies go from how do we isolate these EVs, and here in the institute there's a big focus on scalability of these vesicles. So how can we already have protocols and ready for isolation that are industrially scalable. And here we have a process for isolation of EVs from whey, which is scalable to the 100,000 liter scale. And this really gives us the opportunity to have large amounts and then really go into the clinics with it. And we are also looking into different storage forms, and this process for the isolation of whey EVs is also patented. Another topic is EV engineering, and which involves, for example, targeting or labeling, which I will talk a little bit more about later. And we are also looking into loading strategies. So here we have also recently published a manuscript where we compare different hydrophobic or cholesterol-tagged small molecules and see how they can be loaded into vesicles. And we have just recently submitted also a manuscript on hybrid EVs, where we work together with a group at the Mendel University. So if you're more interested into this, you can look into these applications today. However, I will focus a little bit more on the characterization of EVs and what technologies we can have for that. So as I said, we have a broad array set up here in the labs, which is mainly bulk characterization, like Western blots, protein content measurements, which is quite a basic characterization. We also do analytical SEC and different omics or infrared spectroscopy. But today I will focus a little bit more on the single vesicle characterization approaches, like, for example, with electron microscopy or single vesicle imaging. Another approach is single molecule spectroscopy. So I think we all know why single vesicle characterization is very important. So it's for once we know that vesicles are very heterogeneous, and we have very many different EV subpopulations that can tell us a lot about the cell type, about the cell state, and it really requires the characterization of these different EV subpopulations. Also, with different isolation methods that are used within the field, we know that we isolate different populations of EVs. And to understand and to compare different studies, we have to know what, with our isolation method, what we really isolate and what subpopulations of EVs we have. So I think we all agree it's very important to do single characterization on the single vesicle level, and this can be done with different methods. I think one very prominent method that is used a lot in the field is flow cytometry with nanoflow instruments, which sometimes also have imaging capabilities. And here the field is already has already published a lot of things that, to compare these different instruments, and also to make the people aware of what controls are needed and everything. Another method is to image the vesicles after capturing them on chips, which is, for example, done by this Oxford Nano imager. Here you see an example of images that were taken with this instrument. It uses first the capturing on the chip, and then you image those vesicles with super resolution microscopy. And of course, you can also look into electron microscopy, where you also have single vesicle resolution, and you can characterize them. What all of these methods have in common is that you either required some very dedicated instrumentation, or you need some proprietary chips to immobilize them. So it's always connected to some costs. However, we and others have shown that single vesicle imaging can also be done by using only standard low-cost lab equipment. And the only thing that is really required for doing single vesicle imaging is you first have to immobilize your EVs on a slide or somewhere, which can be done on regular microscope slides. Then you have to image them, and imaging can also be done with regular wide field or confocal microscopes to get images like you can see it on the top here. And then you have to somehow analyze your images. And this is where we found to have a gap in the field, and also driven by our own need, we started then to develop EVAnalyzer that should help us with this automated image analysis. So what is EVAnalyzer? EVAnalyzer is or EVAnalyzer, as we started, was an image-J plugin, which has pipelines which were optimized for EV research. And we were able to do this because we had Joachim Dannmeier, a programmer and software engineer, joining our lab, and he helped us with this. So the tool should enable you to analyze your images at high throughput and in a batch, and it should enable you to do single vesicle quantification on our images. So here we can count our AVs, we can co-localize different fluorophores to see which markers we have on, and we can co-localize up to 5 channels. And we can also automatically remove reference beads. Reference beads are sometimes added because then focusing is easier. You don't bleach your sample. So it's sometimes nice to add these reference beads, and they can be automatically removed from the analysis. Additionally, we also have pipelines there for single vesicle quantification, so to really quantify the vesicles in a cellular context. What you get as an output is, first of all, you get control images, so you see what was analyzed, and you also get an Excel output with all the data from your images. So you have the data from the mean data of the different images, and also you get information on each single. Additionally, what we did is we have looked into immobilization of EVs and also into immune staining of EVs. And we have optimized the protocol for immune staining on EVs. So we saw that EVs can nicely be immobilized on normal microscope slides. You just have to incubate them for 30 minutes. Then they can be fixed where formaldehyde works best for us. We can do blocking, and here, for example, normal goat serum works very well. And then we tried different embedding media, which then helps you to really maintain your fluorescence. And here, for example, Vector Shield worked very well for us, but also others can work well. So this plugin, EVAnalyzer, and the optimized protocol we published some years ago in JEF. We have also shown this at the EV Club, and since then we're really happy that we have continuous downloads of the image of this image-J plugin, and people are more and more using it. Today, I want to give you some application examples. Some of them were in the publication, some of them are new, and then I want to tell you a little bit how the tool has evolved over the years and how you can use it. So I think the most prominent, the most important application example for single vesicle imaging and for the EVAnalyzer is to validate labeling strategies. So labeling generally can be done either by genetic manipulation of the producing cell. So you can manipulate your cell, for example, to have a tetraspanine with a GFP on it, and then your cells will produce EVs that have a tetraspanine with a fluorescent tag. Alternatively, you can also go for post-isolation labeling. Here, a big benefit is that you're not restricted to sources where you can do genetic manipulation of the producing cells. So here we can go for any of the different cell types, also for edible sources. And also a benefit is that you have a more generic label of different vesicles because you usually don't label one specific protein, but you label components of the vesicles like proteins, lipids, or carbohydrates. And when we started in the field, we wanted to find labeling strategies for later than tracking our EVs, and we first systematically validated different EV labeling strategies and to see which one works best for us. So what we took is we took CD63 neon positive EVs or GFP positive EVs, and then we used these different post-isolation labeling methods to see if they really label our CD63 positive EVs. And there are many different lipid ties or available on the market. And what we saw is, for example, for DII labeling in our hands, we didn't get any co-localization. So the DII, we get some spots, but they do not label our CD63 positive EVs. For other strategies like calcein labeling, we get only partial labeling of our EVs, but not all of them are labeled. So this is also not an optimal dye. What worked best in our hands in the end was cholesterol anchoring, which you can see here. So what we have is a cholesterol which integrates into the membrane. And then we have a spacer and where we have tagged TAMRA, which is a fluorophore, to it. And as you can see here on the bottom, we get a very bright and nice labeling of our CD63 positive vesicles. So we really know that this label is really attaching to the vesicles. Another strategy that works very well is covalent labeling of primary amines, so labeling of proteins. And also here you can appreciate that there is a lot of co-localization between the TAMRA NHS, so our fluorophore, and the CD63 neon positive EVs. So we had two labeling strategies that work very well for us, but before going into in vivo studies or also into in vitro cellular studies, we first wanted to make sure that the label also stays with the EVs. So some labels have the problem that they might shed off the EVs. And what you can see here is an experiment where we had our EVs, our labeled EVs in PBS or in serum, and we see that if we label them with the cholesterol anchoring, as soon as we put them in the serum, the label sheds off. While with the NHS labeling, with the protein labeling, we have a nicely stable labeling of the CD63 neon positive EVs, and which is also very stable in serum. So I think the take-home message here is that I think it's really important to invest the time to really characterize the vesicles in deep and make sure that you label the right thing, and also that the label stays with your vesicles if you want to track them in the cells or even in vivo. Another application example is for immune labeling and antibody validation. So within the publication, we also have a protocol for immune labeling, and we are using this a lot for antibody validation. So what we take is, for example, HECIVs that produce or that were transfected with the CD81 emerald GFP, and these cells then produce vesicles that have a CD81 with a GFP tag. And then we use different CD81 antibodies to see if the antibodies really label our CD81 positive EVs, which in this case worked very well. These validations can be done either on isolated EVs, but they can also be done directly in conditioned medium. So also here we get a very similar result. We see that the label works well, and this is really enabling because you can do the immune labeling directly in conditioned medium with without any further isolation before. Additionally to these parameters that I described now, like colocalization and the count of the vesicles, we also get out additional parameters of the vesicles, like size, circularity, or spot intensity. Here, one has to be cautious that size in this case is not real size, because we are imaging those vesicles with standard wide field microscopes, where we have a resolution of approximately 200 nanometers, and so we cannot resolve real size, but the size is very dependent on how bright these vesicles are. So what we get out, though, is how bright these vesicles are. And to showcase this and to look at this parameter, we took differently engineered cell lines, which produce vesicles with different or which produce differently bright vesicles. So we have a CRISPR line which has very dim vesicles, or a line where we transfect CD63 with a fluorescent protein. And so we have these different brightnesses of vesicles, and then we analyze them either with fluorescent correlation spectroscopy, which is a method where we can look at molecular brightnesses, and so we get the number of fluorescent molecules per vesicle, or we analyze it by single vesicle imaging and analysis with EVAnalyzer, and we get these spot intensities. And what we saw is that they correlate really very well, so we can correlate between the brightnesses of the vesicles and the number of fluorophores that are on these vesicles. The benefit of single vesicle imaging here is that you get the brightness for each individual vesicle, while with the fluorescence correlation spectroscopy, you only have average molecular brightnesses of your EV samples. So to really calculate that back, the single vesicle imaging data into numbers of fluorophores, we were looking for calibration beads. And I have to say, this took me very long to find a company that produces these calibration beads. And only recently I find this Quant company that produces these small fluorescent beads, and you can order them with different number of fluorophores. So what we did is we ordered these Alexa Fluorophore 88 beads with 10 to 200 fluorophores per bead, and then we looked at it with single vesicle imaging, and we can see that we can nicely separate the different populations of EVs, and we can generate this nice calibration curve with our fluorophore. This really enables us now to correlate a vesicle brightness from single vesicle imaging with the number of fluorophores. We can compare the brightnesses on different instruments, on different microscopes, in different labs, and we can do absolute quantification of labelings, but also of loading quantifications. So what we did with it is we, for example, looked at imaging with an 100x objective on microscope slides. In this case, we had the Sci5 fluorophores in the beads with 20 to 200 fluorophores. And then we imaged the same thing on different plates, on IBD plates. And also we did the imaging with a different objective, with a 40x objective. And when we compare this, we can see that with the 100x objective, we get similar results. So we can easily change between the IBD plates or the microscope slides. But with the 40x objective, of course, the vesicles are much dimmer. The brightest vesicles are only approximately 3 times more bright than the background. So we lose a lot in resolution when we go for the 40x objective. But it's still possible to distinguish these 20 fluorophore beads over background. So one example what we can do with it, I will show you here. These are LNPs, and we have a Sci5 fluorophore in the lipids of the LNPs, and the LNPs are loaded with siRNA, which has an Alexa fluorophore 88 tag. And with this, we can, for once, also look at how many of the LNPs do have an siRNA loaded, which is approximately 20%. And then with the additional information on vesicle brightness and the calibration beads, we can also calculate back that we have approximately 40 Alexa fluor for 88 fluorophores per vesicle. And we can also look into the single vesicle data. So we see here the distribution of the single vesicles, and an average vesicle has approximately 20 of these Alexa fluorophore 88 fluorophores, while there are also some that have more than 100 of these fluorophores. So there is quite some distribution of how many siRNA is loaded into these LNPs. And we can also look at the Sci5 brightness and get information about the size of these vesicles. So when we, for example, look at the Sci5 samples and we look at loaded particles and the non-loaded particles, we see that the loaded particles are much brighter. So there is more lipids in there, so they are bigger. So loading of the siRNA is mostly seen in these bigger LNPs. So I think the important thing here is to know or to take home is that there's a lot of information you can get with this sort of analysis, because you really get information on each vesicles and with the calibration beads even on how many fluorophores you have per vesicle. Single vesicle imaging can also help, for example, in cellular assays, which are, for example, set up here in the lab by my colleague Patricia, where we look at a cargo monolayer with mucus-producing cells, and we add our EVs and then quantify how much of the EVs are transcytosed, and then we analyze our EVs in the basal layer and to see if transcytosis is happening. Another example is we started during COVID to work also on antiviral EVs, and we produced EVs with an S protein or an ACE2 on the surface. And as when you have a fluorescent tag on your proteins, you can also quantify how many EVs do you have with an S protein, how many EVs do you have with an ACE2, and you can really go into this characterization. From going from single vesicle characterization to tracking the EVs, we also want to see, okay, where do they go in cells, for example, and here we are already building on quite some knowledge from Nicole Meisner-Koper's old lab at Novartis, where they already did a lot of live cell imaging and EV tracking. And they were one of the first ones to show that exosomes serve from filopodia and they are taken up by the cells at endocytic hotspots, and they are taken up as intact vesicles. So in order to be able to track the EVs in a cellular context, with EVAnalyzer, we have also dedicated pipelines for EV detection in the cells. So this is a bit requires a bit more filtering, because there is a lot, or there's more autofluorescent background within cellular and the cellular environment. Then we also have pipelines for cell detection, which also works on bright field images, and we have pipelines for nucleus detection, which helps us to separate the cells. So as you can see here, we use the nucleus to form this Voronoi grid to then separate our cells and get information about vesicles or spots in single cells. So when we start with an image like this, where we have A509 cells, which were treated with milk EVs, which were labeled with Cy5, which we can see here in red, then we can analyze this and get an output image like this, where we see how our cells were separated, which vesicles are detected, and this can be done nicely with these pipelines. The information you can get out is you can get the mean fluorescence per cell. You can also get the mean number of fluorescent spots per cell, and the cell area, how big are your cells. You can have the EV numbers for each individual cell, so you can make correlations like here, that you look at if bigger cells take more cell EVs up, and also you can look at the brightness of the EVs, of course, and you can also do colocalization outside the cell and within the cell to have colocalization between different fluorophores, between different EVs, or between EVs and different cellular markers. In my last part, I want to tell you a little bit about the biodistribution of EVs. So this is also one topic. So after characterization and tracking them in cells, we also want to know where they go in a living organism. And for this, we have set up pipelines that we label our EVs, and then after different routes of administration, we look at whole organ epifluorescence, which we can see here. Then we also quantify EVs in serum, so we can know what is the pharmacokinetics of our vesicles. And we are also looking into histological sections. Here, for example, you can see a liver, where we have in red our EVs, and in green we have a cell-specific stain, so we can really see where in the organ the vesicles are and in which cells they specifically go. For these advanced analysis and quantifications, we have we hit the limits of ImageJ, of our ImageJ plugin. So a few years ago, we started to look into other ways how we can quantify this. So we are limited here in image size. So these big histological images cannot be quantified with the plugin anymore. And we have these predefined pipelines, which do not give us too much flexibility. So what Joachim did then is that he moved different image processing tools that were programmed in Java that ImageJ uses, and he transferred them to C++ programming and transferred them to ImageC. The benefit of C++ versus Java is that it has much better performance, it uses less RAM, it has lower energy requirements, and it can process faster. So for this kind of applications where we have the huge images, we need all of these. And additionally, all the pipelines from EVAnalyzer were also transferred to EVAnalyzer 2, so everything can be used as it was with EVAnalyzer 1. What we can do with this now is we can quantify these vesicles in liver sections. Here, for example, we have CD63 neon EVs that were additionally labeled with Cy7, and we can see that we can find back our EVs with the Cy7 label, and we also see they still have the neon fluorescence on. So we really have CD63 positive vesicles that we find in our tissue. Apart from quantifying these huge images, we also get information about the spatial localization of these EVs. So here, for example, again, we have a liver section, and what the program does, it splits the image into tiles, then automatically processes the single tiles, and then puts together the image again. So what we get here is we can generate these density maps and see if there are some areas where we have more vesicle or less vesicles. Here in the liver, it usually is quite homogeneously distributed. This can also help us with looking at different well formats. So if we have experiments here, for example, in the 384 well plate, we can generate these heat maps to see on one glance where there are more vesicles or less vesicles taken up, and we can look into the individual wells, and we can see if cells or vesicles are homogeneously distributed within the well, because we don't lose the spatial information. Additionally, with the new EVAnalyzer 2, we have very flexible pipelines. So first of all, we can not only use thresholding, but we can also use AI for object detection. We can classify our objects to different object mass, and then all of the data is stored in a data storage, which is then exportable with either Excel or R, or in these user-defined heat maps. And with this, we can also now quantify and detect vesicles in serum, for example. So here you see exemplary images of serum, and in Cy5 and Cy3 we have our vesicles, and additionally we get this fluorescent background. And with these new flexible image pipelines, we can just subtract the background, which we take from a channel where we don't have EVs, and then just detect our EVs. And with this, we can detect and quantify serum EVs directly in serum, and with the small well formats, with the 348 wells, we only use approximately 20 microliter or 60 microliter, I think, of serum for that. And with this new pipeline, we can also go into AI detection of objects. This can be done either with pixel classification. Pixel classification means you annotate your images yourself. Here is an example where we have some cells annotated and background in black, and then we train, for example, a random forest tree, and then get these results. So pixel classification can really be done with a low amount of annotations, and you get quite good results for these easy tasks like detecting the cells. Additionally, or the other possibility is object classification. Here, you really need to train a lot of data to get these results. And we have trained our own AI model, but only with approximately 100 images, and it works quite okay. But here, there is a lot of models already published and out there, which are also freely available. And one is CellPose, which works very well for cell segmentation. And we also implemented this into EVAnalyzer 2, so it can be used for cell detection. Last, this is what I want to show you is the graphical user interface of the new EVAnalyzer 2. What you see here is For example, here is our section that you have seen before, our liver section, and you can navigate easily through this section in the program. And what we do here is we detect the nuclei. So this is a pipeline which we had also produced and programmed for EVAnalyzer 1, where we detect nuclei, and you can see at one glance how many nuclei are detected within this area. You have every nucleus as a single region of interest, and you can click on one nucleus, for example, and you get the information about its size, its brightness, and everything. So the user face is really easy to use, and yeah, I think it's very useful for any kind of image quantification. To sum this up, we started the journey of single vesicle characterization with our EVAnalyzer 1 tool, which is an open source and open access tool, and which was programmed for analysis of a lot of images, and also it supports a lot of different image formats. You can find the information about this in this publication. Also, you find there the protocols for immune labeling and immobilization of the EVs. With going into in vivo applications with the bigger images and with the more complex samples like serum samples, we switched to EVAnalyzer 2, where we have the flexible pipeline creation. We can use AI, and we have a very nice live preview of the samples. This EVAnalyzer 2 is not yet published, but it's already available. So you can just go on evanalyzer.org, and you can find there the download of the software. You can also find the documentation, how to use it. And so it's already freely available. And if there is someone who also wants to join the programming or want to contribute an image processing or analysis pipeline, also always feel free to reach out to us and to Joachim. He also has a page on GitHub where all of the code and the EVAnalyzer can also be accessed and downloaded. With this, I want to thank the group, of course, and especially, of course, Joachim, who helped us a lot with the image analysis and programming. And I also want to thank you for your attention, and I'm happy to have a discussion now.
Hello, thank you, Dr. Schürz, for your presentation. We have a couple questions ready. But if anyone wants to add more, please go ahead and do so in the chat. So the first one is, what is the most reliable bead-based flow cytometry strategy for identifying A549-derived exosomes, including capture bead selection, exosomal marker detection, fluorophore panel design, and instrument setup?
Okay, I have to say I'm not at all a flow cytometry expert, so I cannot answer this question, but I know there's a lot of publications where they compare the different instruments, and also where they compare the panels. I think there is a lot of data out there on that, but I'm unfortunately not the person to answer this, because we, as said in the presentation, we do not do flow cytometry, but we mainly work with single vesicle imaging.
Okay, have you used any beads for your microscopy that could also be used for flow cytometry?
So yeah, so we have, as I mentioned shortly, we have for focusing with microscopy, it's sometimes useful to have some beads, we use tetraspec beads, so they have fluorescence in all different channels, so you can always spike them in and just focus your sample in a channel where you don't have your EVs, and then you can make the image directly on your EVs without focusing, without bleaching them. This is especially important if you want to quantify the intensity and the brightness of your EVs, because then you don't bleach your EVs, and you know, okay, it's really, you have focused on these tetraspec beads, and you just have to additionally take one channel where you have only the tetraspec beads, so you can tell the program, okay, my tetraspec beads are there, and please remove those in the other channels because the fluorescence is everywhere. So this is for focusing, which is very useful. But for the calibration, they don't work because they don't provide any number of how many fluorophores you have on the beads. So for this, it was really tricky because there are some beads which they also use in flow cytometry, but here also either it's so many fluorophores that are on one bead that you are not in a range where you would be with EVs, or you would have, or for a lot of companies, they don't give you any numbers on how many fluorophores there are. They are available in different brightnesses, but not really to calculate the numbers. So for this, it was really only these scatter quant beads that I found so far.
Okay.
What I want to
Sara.
add for the Gata Quant page, just they are quite expensive and they are not very long for storage. So you have to use them quite quick. But if you use the same instrument for the single vesicle imaging, you can make this curve and then just use it. But they are unfortunately not very stable very long. Yeah.
Okay. All right. You mentioned that there's a specific range that you can detect for the fluorochrome to protein ratio, but can't detect. What is the range?
You mean
Is
how
it different?
for the fluorophores, you mean?
Yeah,
Or
is the range different between microscopy and flow cytometry, like how much you can detect?
I'm not sure, to be honest. For the microscopy, so for the one that we have, we are very sensitive, and I think this is the case for a lot of microscopes. We are very sensitive in the blue-green area. So with GFP and this Alexa Fluor 488, we are very sensitive. So we get very bright spots even with only 10 fluorophores. We haven't tried down, so lower, because as I said, they're quite expensive, but I think we can detect particles with 2-3 fluorophores maybe, while in other channels, like we also work a lot with near infrared dyes like Psi7, Psi5. Here most instruments or microscopes are less sensitive, so here we are in the range of 20 fluorophores to 50 fluorophores even, so that we can detect them. I think for flow, as far as I know, it's the differences are not so big maybe, but I think, yeah, I think with the in the blue channel you cannot get much more sensitive than this. I'm not even sure if you would be able with some microscopes to go for really single fluorophore detection. I'm not sure, but yeah.
Okay, thank you. The next question is identifying EVs using markers at the tissue and cellular levels is truly remarkable. I have never seen it before. Could you please highlight it a little more? How did you do it?
Mhm. So I think I I think it's about the histological sections and how we see or we quantify the EVs there. So I mean, first of all, we have worked a lot on the labeling, as I said. So I think it's really important if you want to track your EVs after going in vivo. So we have to have really bright fluorophores on it to be able to detect them. Also, what works very well for us is going into the near infrared region, because here you have not so much autofluorescence from the tissue itself. So you get quite low autofluorescence, and so it's easier to detect these spots. And then for the staining, it's mainly regular stains. So what we do is cryo sectioning of these different organs, and then this finding these EVs is really due to the labeling that we did before.
Okay. So Nebula has a question. Thank you, and a comment. Thank you for sharing your interesting research. You have tracked EV's distribution for months, as I understood from one of the slides. What about EV stability and this duration? Do we need some special points to follow?
So I think I didn't get the question, but I think it's more that I didn't that the sound was not good. So
Oh,
I followed EVs.
yeah. So she's saying that you tracked EVs over months of their biodistribution patterns over time, but
As
she wants to know how you measured their durability over their stability over this time, and if you have any tips on how to maintain their stability in testing over time.
Mhm. Okay, so for the biodistribution, it's a very short time that we looked at. I mean, we for the liver, we had a 5 minutes time point up to 24 hours time point, and we see that especially for the liver, so which is the organ where we have the first where the EVs go first. If we, for example, inject IV, then we have quite a high signal at the beginning, which declines then after 24 hours. So it's not monitoring them over a month. I hope after a month they are gone completely. Um, but so for this, it's really the short time points, and also when you look at them in serum, the serum levels decline very fast, and this is something that also others have shown a lot already, that you have a peak first and then they decline very fast, and then they really go into the organs. So here it's really about not more than 24 hours where we track them, and there are organs where they come a little bit later, like kidney, but they shouldn't be too long in the circulation. And for storing them outside, I think there is also some data that we have how to store them. I think what works very well for us is storing them at minus 80 degrees and really snap freezing them, where they usually stay quite stable over a longer time. So if this is of interest.
Okay. And a question by Vania. Thank you so much for your presentation. In your loading efficiency data, the frequency distribution shows two distinct populations, loaded and non-loaded particles. Can EVAnalyzer automatically separate and quantify these two subpopulations based on fluorescence intensity thresholding?
Mhm. So yes, with the new tool, you can automatically separate them. So with the new tool, the goal was to have, okay, you tell it what subpopulations you want to have. You say, okay, I want to have the vesicles that only have the Cy5 and not the Alexa Fluor 488, which would be the non-loaded. And you want to have those that have the double label, and you separate them and say, okay, I want all the parameters for this population and all the parameters for this population. So yes, you can automatically sort it, and you can filter them by different things. You can say, okay, I want only the ones that are very bright in these populations, or that are very small, So you can do this automatic filtering, which was not possible for the EVAnalyzer 1. I mean, you still get the data, because you have them in the Excel. You can sort them by yourself and just say, okay, do they have what they have, but then you have to do the work in the Excel. So with the next generation, it's much easier because you can automatically do the sorting.
That's pretty cool, this filtering technique. I have another question by Bhavan. What concentration of EVs have you used in your in vivo experiments?
It depends a bit. So when setting up these experiments, we have tried many different things, and I have to say I don't know the exact number now that was used for the ones that I showed you. I think it's dependent on what you're looking at. So if we go for EVs from edible sources, we are mostly limited by endotoxin levels, and so we cannot dose more than that. I think we are in the range of 10 to the 10, something like that, for a mouse, but I'm not 100% sure because I didn't do this study. But yeah, it's also dependent, what route of administration. For milk EVs, we are also looking at oral administration, obviously, and so it's always dependent. But I think for IV injections, and I think everything I showed was IV injections, it's in the range of 10 to the 10 per animal, but yeah. Take
Okay,
with caution.
I have one last question, and then I think we can end because it's 4 o'clock, unless you need to leave now. Okay, so thank you for your talk. I've been using EVAnalyzer 1 and have been able to get quite clean results without any background issues at 20x. For counting EVs, would you recommend a larger volume of higher resolution 100x images, or is a smaller set of 20 or 40x shots sufficient?
Oh, cool. I'm really happy to hear that. Also, I have not yet tried 20x, to be honest. I mean, it's very dependent on what microscope you have and what objective this is, because a 20x is not necessarily worse than any 40x magnification. So really cool that you can detect them with 20x. Yes, so as I showed with the calibration beads, so the difference between background and your beads or your EVs is much taller when you have higher resolution. So you can have a much nicer separation of different brightnesses, because if you are already at the border, you have EV or no EV, but you cannot tell anything about the brightnesses. So I think it's dependent on your question, of course, but yes, high resolution always gives you more of resolution, also in these brightnesses and the different populations.
Okay, thank you so much for your presentation. I'm going to go read your manuscript now about EVAnalyzer. It sounds pretty cool. I haven't
Thanks.
used it
So
yet.
thanks for the invitation, and if anyone
you
has
so
still question, they can also text me, and yeah, if there are problems with using it, we are always happy to also help.
Thank you, Melanie, and thank you, everyone. Thank you, Heather, and we'll see you in the next Snap Talk. Bye bye, everyone.
Bye.